Hi everyone, in this video we are going to talk about the final step of cellular respiration, which is called the electron transport chain. So this occurs within the inner membrane of the mitochondria, and it is going to require oxygen. Oxygen, and I'll show later in the video, is going to be the final electron acceptor. So let's review the anatomy of the mitochondria really quickly. You will see that I've got the outer membrane already drawn in. This green membrane that I've started is the cristae, and here where I have this bracket, I'm going to go in and draw the enzymes that are going to be involved in this reaction. So I've shown the bracket to kind of represent that we are blowing the membrane up at that portion so we can see what's going on there. And that is called the cristae. Then the space here between the two membranes, so between the outer membrane and the inner membrane, is called the inner membrane space. And then all of this area, if we were to fill this in here, so inside of the inner membrane, is known as the mitochondrial matrix. Now, the whole reason that the mitochondria needs two membranes here is because we have molecules of hydrogen, RH+, out here, and we need to move those into the inner membrane space. So if you think back to the light reactions of photosynthesis, we had to move hydrogen molecules in order to make ATP, and the whole goal of the electron transport chain is to create a high yield of ATP. So this is going to be the most efficient, highest yielding ATP producing step out of anything in photosynthesis or cellular respiration. So the enzymes that are involved here are called cytochromes, and you don't actually have to know their names, but I'm going to go ahead and draw them in here. So you've got your first cytochrome, which is technically called NADPH reductase, and then you've got another one in here which is going to be responsible for picking up your FADH2 molecules. And then the other ones are super generic. We really don't need to know their names at all. They're going to be involved in moving electrons and exciting the electrons so that they can pump hydrogens across the membrane. Okay, so now I'm going to go in and fill in the lipid membrane here. Okay, and now because we're going to be making a whole bunch of ATP, we need to add in a molecule of Atp Synthase. And then we'll go in and fill in some more of this membrane here. And then close up the membrane. Alright. So I'm going to erase this label, the matrix, so I have a little bit more space to work on the inside here. Now we're going to start representing the movement of the electrons. So remember, we have these electron carriers that are produced during glycolysis, during the prep step, as well as during the citric acid cycle. And remember, if you think back to your yields, we make 10 total molecules of NADH. So all 10 of these molecules are not going to drop off their electrons at the exact same time, but just realize that we do have 10 NADHs from each cycle of one molecule of glucose. So I'm going to write 10 NADH here as a reminder, but realize not all 10 of them are going to drop off their electrons at the exact same starting location, okay? But all along the membrane there exists these different cytochromes. So that is going to get kicked out as NAD plus as it's oxidized, and then we are going to drop off our electrons to our NADH reductase. And remember, when we have these electrons getting dropped off, their whole purpose is to pull H pluses down here. So the H pluses that were broken off from your NADH, notice NAD plus does not have those H's. So those hydrogens are pumped down into the inner membrane space. Then our electron is going to take a little trip, and we're going to go to the next molecule here. And this is where we are going to have our 2 molecules of FADH2. So they're going to be dropping off right here. Remember, they also store electrons. So we're going to end up with 2 molecules of FAD. So they also store electrons. These ones are going to be involved in energizing the electrons once more so that they can move to the next cytochrome and pull some more molecules of H plus down here. So now we're getting all sorts of H pluses building up down here. Okay, the electron is going to move on its way, and it has just enough energy left to pull one more molecule of hydrogen into the inner membrane space. Okay, so now those electrons need to be picked up, and we already know that water is a byproduct of cellular respiration, and so that is going to occur at this stage here. I'm going to draw it over here since we're kind of running out of space. But oxygen is the final electron acceptor, and the way that that works is you're going to have 2 molecules of H plus that are in the matrix. You're going to have your 2 electrons, so those charges are going to cancel out in 1/2 of an O2. So remember, oxygen exists as O2, not as oxygen itself. So we're going to use 1/2 of an O2, and that's going to produce 1 molecule of H2O. So we end up getting 6 total molecules of H2O being produced for every molecule of glucose. Okay, so now we've dealt with our electrons, they've been picked up and cleaned up, and now we have to deal with our ATP synthesis. That is the whole goal. So this last enzyme here is called ATP Synthase. And remember, just like in the light reactions, all of these hydrogens are going to get shuttled back across ATP Synthase. And so what we're going to get here is our molecules of ADP plus P becoming ATP. And from this process, we're going to get about 30 to 32 molecules of ATP being produced. Now, if you think back to the other reactions in cellular respiration, in glycolysis, we had 2 net molecules of ATP. And then in Krebs, or the citric acid cycle, we had 2 molecules of ATP. So from cellular respiration, our total net yield of ATP then is going to be about 34 to 36 ATP. Okay. So that is the whole entire goal of cellular respiration. So if you think back to the cellular respiration equation, it's just opposite of photosynthesis. So you end up using 1 molecule of glucose and 6 molecules of oxygen, of O2, and that's going to be used here. And then you're going to end up with producing 6 molecules of carbon dioxide. Remember, 2 molecules of carbon dioxide were lost during the prep step, and then 4 molecules of carbon dioxide were lost during the citric acid cycle. And then the other byproduct of this is the 6 molecules of H2O. So you'll end up with a total of 6 molecules of H2O as waste for every molecule of glucose. And then you end up with 34 to 36 total ATP being produced. So let me erase some space here, and we'll review our equation for cellular respiration, since this is the last video in this series. Okay, So when we're talking about cellular respiration, the first thing we need to do is write down our glucose, which is C6H12O6. We're going to use 6 molecules of O2, and that is going to give us 6 molecules of CO2, as well as 6 molecules of H2O. All right, so our carbon dioxides here, these were lost as 2 in the prep and 4 in Krebs, or the citric acid cycle. And those are our 6 carbons right there, and our 6 oxygens. And now we have to deal with our 6 molecules of water here. And that's what we get here, and those are all created as a byproduct of the electron transport chain. Alright, please let me know if these videos have been helpful, and continue watching as I will post more for future lessons as well. So remember, now I skipped this part in the video, so I wanted to make it clear. H plus is pumped against or up the concentration gradient from a low concentration to a high concentration. So since we're going backwards of normal diffusion, this is going to take energy, and that energy comes from the electrons that are excited from the breakdown of glucose. So the potential energy of the higher concentration of hydrogen ions is used to form the bond between ADP and P. And that bond is going to store energy in the molecule ATP once those hydrogen ions flow back down the gradient at ATP synthase. So it's really important to understand that the energy for ATP actually comes from the potential energy that's stored in the high concentration of hydrogen ions in the inner membrane space of the mitochondria.